Hello, we'll be now knowing what exactly is primitive streak. We'll be reading those primitive streak topic under these headings. Like first one, what is primitive streak? Here, this one is the primitive streak. How to write? It is a linear opacity seen in the caudal part of the germ disc, appearing in the third week of the intrauterine life, containing a central depressed region and a peripheral lateral margins. Is it clear? So that is about the primitive streak. Next, formation. How it is formed? Actually, this one is the germ disc which is oval in shape till second week after fertilization. In the later part of the second week, you can see this at the caudal end of the germ disc. There are pluripotent cells of epiblast. Those cells, these pluripotent cells, that means here, this region, this is epiblast, this is hypoblast, this is amniotic cavity, this one is the yolk sac, and this is connecting stalk. So these cells here are pluripotent in nature in the caudal part of the germ disc, that is this one. They migrate, they proliferate and invaginate between the epiblast and hypoblast into a linear opacity or linear streak-like structure, and that is called as this primitive streak. Is it clear now? So from this epiblast, this is proliferating caudocranially to form the primitive streak. What are its parts? Formation and what are its parts of the primitive streak? You can see this is primitive streak. At the cranial end, it gives a knob-like elevation. This one we call it as primitive node or Hansen's node. Primitive node or Hansen's node, and the surface depression within the node is called primitive pit. This primitive node can also be called as primitive knot, and the surface depression is primitive pit. When it appears in the beginning of the third week. It starts appearing at about 15th day after fertilization and becomes complete. It grows caudocranially, becomes complete by the end of the third week and disappears by the end of the fourth week. Functions. You need to remember, number one, it is a primary organizer of the body and induces the formation of intraembryonic mesoderm and notochord. Number two, it gives rise to intraembryonic mesoderm. Number three, it gives rise to notochord. Number four, it demarcates the body into two equal halves. So it divides the embryo into right and left halves equally. Number five, it represents the craniocaudal axis of the embryo. So these are the five functions you can write for primitive streak. Next, fate. What is the fate of primitive streak? It appears at 15th day. It disappears almost completely by the end of the fourth week. And the disappearance is craniocaudally, it disappears. And some cells may remain in the embryo as an insignificant structure in the sacrococcygeal region of the embryo. In the sacrococcygeal region of the embryo, some remnants of this primitive streak may be present as an insignificant structure. Clinical embryology. Those cells which are remaining may proliferate because they are pluripotent and may give rise to sacrococcygeal teratoma. Those primitive streak cells which are left over may proliferate and may give rise to sacrococcygeal teratoma, which can turn big, may cause obstruction in labor, may cause the death of the baby, and it is one of the commonest embryological tumor of the newborn. Incidence is 1 in 37,000 pregnancies. It is most commonly seen in females and usually turns malignant, initially benign but turns malignant, and should be removed within 6 months after birth. So this is all about a small topic called primitive streak, which is a important question at the university level. Thank you.